Pittenger, and I am a Senior Applications Scientist for the AFM business at Bruker. I would like to welcome you to this installment of the Bruker AFM webinar series. This presentation is titled AFM and Cell Mechanics, Investigating the Nanomechanical Properties of Living Cells, presented by Dr. Andrea Slade and Professor Manfred Radmacher. Before I introduce Andrea and Manfred, I would like to make a few quick logistical comments. First, we encourage your participation during the webinar, and if you have a question, please type it into the questions dialog box on your screen. We will accumulate these questions throughout the presentation, group them together, and then answer them at the end of the prepared remarks. Quite often, we will have more questions than we can answer in the session, and in those cases, we will be sure to follow up with answers via email afterwards. Also, if you would like to review the presentation afterwards or pass it on to a colleague, the webinar will be posted online within 1 week in the webinar section of the Bruker website. As a follow-up to this presentation, you will also be receiving an email with this link. Finally, when you exit the webinar software, you will be asked to take a brief survey. We would very much appreciate it if you would take the time to complete this, as it helps us pick the topics that are most important to you and to generally make this series better. So let's get started. Let me introduce our presenters. First, Andrea Slade has been an application scientist for Bruker for 11 years, focusing on life science research. Professor Radmacher studied physics at the Technical University of Munich. After a postdoc at the University of California, Santa Barbara, he returned to the University of Munich. In 2000, he moved to the Universität Göttingen for his first faculty position. Since 2002, he is professor of biophysics at the University of Bremen. His current research interests are mechanical properties of cells and their relation to diseases. He has published 66 papers, 6 book chapters, and 2 patents. So let me turn it over to Andrea to begin this talk entitled AFM and Cell Mechanics, Investigating the Nanomechanical Properties of Living Cells.
So thanks, Bede, and thank you to everyone for joining us in the webinar today. For the first part of today's presentation, we're going to discuss the latest advances in Bruker's Bioscope Resolve AFM and how they're enabling new avenues of research in cell biology. Bioscope Resolve is designed to be integrated with an inverted light microscope and is compatible with advanced optical microscopy techniques such as confocal laser scanning, as well as super resolution techniques like STED and PALM. Using Resolve's MoreauView software, optical images are fully registered to the AFM scanner and can be used to locate and target specific locations. One second, sorry. I apologize, I had my screen paused there. Okay. So continuing on. Using Resolve's MoreauView software, optical images are fully registered to the AFM scanner and can be used to locate and target specific regions of interest on a sample on which to conduct AFM measurements. Resolve also provides synchronization of both AFM and optical data collection, which includes automated acquisition and import of registered optical images directly into the MoreauView software. With peak force tapping, Resolve provides the highest resolution imaging capabilities for any BioAFM, whether it be studying small individual biomolecules such as proteins and DNA, or imaging larger bacteria and mammalian cells. And the Bioscope Resolve also offers the most comprehensive package for studying cell mechanics. Not only does Resolve have the proven benefits of peak force QNM and fast force volume modes for investigating the nanomechanical properties of cells, we have now added ramp scripting capabilities, which we'll get into further detail in this presentation, which now allows researchers to conduct advanced and customized studies of the viscoelastic behavior of living cells. In peak force tapping mode, the AFM probe is modulated at a fixed frequency. As the probe is brought into contact with a sample surface, the feedback signal is the maximum force or the peak force between the tip and the sample. Peak force tapping uses sinusoidal ramping. As such, the tip velocity approaches zero as the tip approaches the sample surface. And because peak force tapping uses a continuous feedback loop, it is constantly detecting the peak force. This enables peak force tapping to directly control the tip-sample interaction force, which facilitates the use of low imaging forces even when operating in fluid. This helps protect both the probe and the sample from potential damage or distortion and is really the key to enabling high-resolution imaging. And because we're essentially performing thousands of force curves per second, peak force tapping images can be acquired at traditional imaging mode scan rates with standard pixel resolutions. With peak force tapping on Resolve, we've been really able to push the limits of high-resolution imaging on biological samples. In the top image on the right, we have a movie showing real-time scanning of plasmid DNA. All over the DNA strand, we're able to resolve both the major and the minor grooves of the double helix, which are 2.2 and 1.2 nanometers in width. And the resolution remains very consistent in repeated scans, with over 15 minutes of continuous imaging. And when it comes to imaging live cells, peak force tapping has enabled some really nice breakthroughs in high resolution. In the lower image, Schillers et al. Used peak force tapping to resolve the structure of microvilli for the first time on living cells by AFM. And because peak force tapping provides direct control of the imaging forces, they were actually able to establish a direct relationship between the observed structure of the microvilli and the interaction force of the AFM probe. Peak force tapping has also enabled peak force QNM, or quantitative nanomechanical property mapping. Because we're essentially collecting force curves at every pixel of a peak force tapping image, we can use these individual curves to obtain quantitative maps of the nanomechanical properties of cells that directly correlate to the topography data channel. The peak force QNM images shown here are of live MDCK cells obtained on the Bioscope Resolve, with the topography image on the left and the corresponding modulus image on the right. The color scale in the modulus image corresponds to increasing modulus values, with darker colors having lower modulus and lighter colors having higher modulus. Keeping this in mind, when we look at the modulus image, we can see long, thin structures corresponding to actin fibers that appear to have higher modulus, being lightest in color, while cell surface features, which we believe to be microvilli, appear softer or darker in color than the cell membrane itself. Now, a rapidly expanding area of research is mechanobiology, which focuses on how cells sense and respond to forces in their surrounding environment, as well as how these forces affect the mechanical properties of cells. Examining the effects of forces on cells has a wide range of applications, from understanding disease pathology to the development of tissue engineering devices. So when it comes to studying cell mechanics, the combination of force spectroscopy, fast force volume, and peak force QNM on the Bioscope Resolve not only provides the widest range of operating frequencies from about 0.1 Hertz up to 2 kilohertz, the overlap in frequencies between techniques allows easy comparison of the data obtained from these modes, as well as the opportunity to study frequency or time-dependent behavior. Bruker's unique peak force QNM live cell probe has a low spring constant, a very tall tip with a fixed end radius. Having a well-defined tip end radius helps in fitting the various models for elastic modulus calculations. The 17-micron tip extends the cantilever away from the sample surface. This actually minimizes the imaging artifacts that often result from imaging tall mammalian cells with probes that have a typical 3 to 5 micron tall tip, and also minimizes the effects of viscous damping when operating in fluid, which actually helps improve the force resolution. Now, probe calibration is also a very important step in obtaining reliable quantitative mechanical property measurements. Peak force QNM live cell probes are individually calibrated for their spring constant using laser vibrometry. With pre-calibrated live cell probes, we can use Resolve's no-touch calibration to back-calculate the cantilever deflection sensitivity, meaning that at no point during the calibration procedure does the tip come into contact with any surface. As such, using pre-calibrated probes not only minimizes error in mechanical property measurements, it also protects the tip or any chemical or biological functional groups on the end of the tip from damage during the calibration process. Another unique feature of the Bioscope Resolve is that it uses vacuum to immobilize Petri dishes on the AFM stage. Rather than using the typical clamps or holders that press against the top of the Petri dish, using vacuum to hold the bottom of the dish provides low noise operation for both high-resolution imaging and mechanical property mapping, while maintaining optical access to the cells. Now, to fully understand cell mechanics, it's not enough to just look at a cell's elastic properties. We need to also consider their viscoelastic properties or time-dependent responses. For these types of studies, the Bioscope Resolve now offers ramp scripting. Ramp scripting provides an easy-to-use interface for designing customized advanced force measurement studies. These measurements can be conducted at a single point or as an array over a specific region of a cell, providing a two-dimensional map of viscoelastic properties. Ramp scripting is also fully integrated with MoreauView, so that optical images can be used to identify and then target specific cells or parts of a cell on which to conduct these measurements. Measurements can also be synchronized with the acquisition of optical images. In the next few slides, I'm going to go through some examples of the types of studies we can do with ramp scripting. In this first example, we worked in collaboration with Professor Manfred Radmacher, our guest speaker today, to conduct studies of the viscoelastic response of live fibroblast cells. Manfred will go into details of the theory behind these types of measurements, but briefly, viscoelastic properties of cells can be quantified by measuring their creep response, or relaxation under force. Using the AFM probe, we apply an instant stress or force to the cell. The force is held constant, and relaxation of the cell is measured as a function of time. Using phase contrast images of the cells in MoreauView, we were able to target the cell nucleus, lamellipodia, as well as the underlying Petri dish surface on which to conduct scripted force measurements. In this particular script, we're holding or maintaining the Z sensor position and simultaneously monitoring the change in force as a result of the cell response. The script uses a double-step hold, where the first 1 second hold allows the cell to reach an equilibrium. We then perform a very quick, small Z step into the cell, hold it for half a second, and then step back out to the initial Z position and hold for another half second. It's important that these movements occur very quickly, as viscoelastic responses of cells occur over very short time scales, somewhere on the order of 50 to 100 milliseconds. And looking at the measurements taken at different locations shown here, we can see that there are differences in the response. Now, this type of study really highlights the advantages of ramp scripting on Bioscope Resolve. We have fast feedback that allows for these very quick movements in Z, as you see here. We were able to move 50 nanometers in Z in about 1 millisecond. High stability enables longer holds occurring over several seconds, and sensitive force control allows us to measure small changes in cell response. And the ability to trigger the acquisition of optical images at the end of each segment in a script enables full integration of cell viscoelasticity studies with advanced optical techniques. Another example of what we can do with ramp scripting is a traditional molecular force clamp type study, where we follow the unfolding and refolding trajectory of a single protein or biomolecule as a function of time. Proper folding of the protein molecules into their three-dimensional structure is critical to ensure their true functionality. Misfolding or mutations that might alter the protein's native structure are often implicated in altered functionality or even disease states. In this particular study, we used ramp scripting to unfold and then refold a single titan molecule. Opposite to what we did in the previous study, here we're actually holding the force and observing changes in the Z sensor. In the first segment of the script, the AFM probe is brought into contact with the surface and held there at a force of 1 nanonewton. After 4 seconds, the tip is pulled away from the surface and held at minus 130 piconewtons for 6 seconds, during which time we observe unfolding of individual titan monomers at repeated step heights of about 25 nanometers, which corresponds to the contour length of a titan monomer. We also observe an overall lengthening in the full unfolded polypeptide chain. In the next segment, the force is lowered to minus 30 piconewtons and held for another 6 seconds. This allows the titan molecule to refold, which follows a two-stage process. Here we have a very fast initial phase, which occurs due to the elastic recoil of the unfolded polypeptide chain, which is then followed by a slow phase with larger fluctuations in the end-to-end length and a decrease in the overall length of the polypeptide chain. Interestingly, the refolding process is not stepwise like we saw during unfolding. In the very last segment, the AFM probe is pulled far away from the surface. Here we see that classic sawtooth pattern that's typically observed in standard force distance curves, where the tightened molecule is being stretched under an increasing linear force and finally breaks free from the AFM tip. While this particular example is looking at single protein molecules, you can imagine that performing this type of measurement on the surface of a living cell and observing the unfolding and refolding of individual cell surface molecules or membrane receptors. In another example shown here, we're looking at the viscoelastic or creep response of bacteria cells by applying a constant force to the cell surface. But instead of conducting single-point measurements, here we combined ramp scripting with fast force volume mapping, such that viscoelasticity measurements are conducted at each pixel in the force volume image. This is helpful, especially in the case of single E. coli cells, which are only a couple of microns in length and a diameter of about 500 nanometers, and so they can actually be quite difficult to target optically. As shown in the graph, we compared the creep response of living E. coli cells to cells that have been fixed with glutaraldehyde, as well as a hard glass substrate. Curves from the live and fixed cells were fit to a standard linear solid model, and the time constants calculated. For the softer live bacteria, the time constant was about 0.92 seconds, while the stiffer fixed cells had a shorter time constant of about 0.33 seconds. We also used our advanced MATLAB toolbox to analyze the data we collected on the bacteria in order to provide new and different information such as viscoelastic property maps. On the left, you can see part of the script written by our collaborator Antoine Desjardins that he used to do this. On the right of the slide, you can see the resulting creep deformation and creep rate maps, in addition to the peak force QNM and fast force volume elastic modulus maps. You can see that there's correlation between the two elastic modulus maps, with both showing softer regions in the cell in the upper left corner. And while the viscoelastic property maps do show variations across individual cells, they are not directly correlated to the elastic property maps and do not show these softer areas on the same cells. Now, in the bacteria experiment, we looked at the creep response at a single frequency, but we can also investigate differences in the response of a cell across various frequencies. In other words, we can actually study the cell response as a function of frequency. In ramp scripting, this can be done by modulating the AFM probe while it's held in contact with the cell surface, and then sweeping through a specific frequency range. The amplitude and the phase of the response enables calculation of both the storage and last modulus. In the example shown here, force modulation spectra were collected at each pixel of a fast force volume image conducted over the surface of a living fibroblast cell. When the measurements performed directly on top of the cell, the resulting spectrum reveals an increase in the amount of deflection with increasing frequency, such that the cell appears stiffer at higher frequencies. Zooming into the area here, we can easily see the sinusoidal motion of the AFM probe. But when the same measurement was conducted on the surface of the polystyrene petri dish, we see no variation in the amount of deflection over the frequency range, indicating that the response is frequency independent and does not exhibit any viscoelastic behaviors. This kind of integration of force modulation spectra with fast force volume imaging creates new kinds of data cubes. If we look at the amplitude response of the cell at lower frequency, we see that there is a large difference in the cell response, marked by regions or points 2, 3, and 4, and the petri dish, marked by points 1 and 5. But at the same time, the response on the cell body, points 2 and 3, and the lamellipodium at point 4 are very similar. But if we now look at the response map at different slices or specific frequencies across the sweep, we see that the lamellipodium rapidly becomes stiffer at higher frequencies, behaving more like the petri dish, while the cell body remains softer with very little change over the frequency range. So to briefly summarize this part of the webinar, the Bioscope Resolve offers a full suite of methods to investigate both the elastic and viscoelastic properties of live cells, including peak force QNM, fast force volume, and now with ramp scripting and force modulation spectra. Together with pre-calibrated peak force QNM live cell probes and no touch deflection sensitivity calibration, Resolve now truly enables easy and reliable measurements for cell mechanics research. So with that, I'd like to now turn things over to Professor Manfred Radmacher, who will present his latest research in using AFM to study cell mechanics.
Excuse me, Professor Manfred, you've muted yourself, so we can't hear you. If you could unmute your phone, go ahead and present.
Okay, so now you can hear me?
Yes.
Am I online?
Yeah, we can
now?
hear you.
Okay, sorry for the technical issue. So I will start from the beginning, right away from the beginning. So it's a great pleasure for me giving here a presentation, a presentation through the internet. It's actually the first time I do that. So that's the reason why I have added here the live webcam, so that you can see that there's a real person talking to you. And also want to encourage you, if there is any questions, you may want to ask them during my presentation, and I will try to answer them. The other questions will be answered at the very end. But I will mute most of the time my video webcam, because I want you to focus on the transparencies. I want to give you an introduction, as Andrea said. I want to give you an introduction today on on how to measure mechanical properties and why to measure mechanical properties of cells in the AFM. Since I'm not sure, I guess most of you are familiar with AFM, but there may be some cell mechanics people who haven't heard anything about AFM. So we'll make a very, very quick introduction to AFM, so that on the chance that there is somebody who doesn't have a background in AFM has a possibility to follow what we are talking about. Just briefly, in the AFM, you have like three major components: a sharp tip, which is mounted on a soft cantilever spring. You can measure the deflection of that cantilever. You bring it into physical contact with the surface, and you can measure the surface topography. For instance, the main point is that the tips are very, very sharp, so that you get high resolution. A lot of people do like imaging of biological samples. You can image single molecules, you can image like living cells. My personal opinion is that the real niche where AFM is unique and will stay unique in the near future is measurements where forces are involved. Andrea was already addressing cell mechanical measurements or molecular interaction forces. I guess this is the real niche, because if you think about resolution, there is competing techniques which are getting better kind of every year, like super resolution optical microscopy. But the combination of being able to measure forces with high spatial resolution, that is something where AFM is still kind of a unique instrument there. Okay, quick outline of what I'm going to present here you today. First thing is, how can we measure cell mechanics? And I want to go into some details of analyzing the data and so on, which will become very important in the later parts of my presentation. Next point is, if we can do that, why do we actually want to do that? So why is cell mechanics interesting? Then comes a parenthesis on how to measure cells' viscosity, because that's a point which has not appreciated widely in the community. Well, there has been several reports by a few groups, but normally people only measure like the elastic properties, but it's actually important to include the the viscosity as well. And finally, kind of as an application or kind of as an example, when it becomes really important to look at the viscous and elastic properties, I will talk briefly about cancer cells. The basic principle of our measurements is shown here in that animation. While the tip is scanning over the sample, at each point we record a force curve, which basically is we are recording the deflection of the cantilever while we are changing the set height. This is a force curve now on the stiff sample where the slope here is equal to 1. The deflection is identical to the set movement. Once the tip will come to the cell, which will happen in a few seconds, you will figure out that the slope here becomes smaller because the movement of the set piezo is kind of separated in deflection of the cantilever and indentation of the sample. From this force curve, we then actually calculate the force being exerted on the cell and the indentation generated on our soft sample. As you can see, this relation is not linear, as you can see here when the tip is back on the cell, because mainly the tip geometry, the contact area is changing. We are using normally tips of a pyramidal shape, and the larger the indentation, the larger the contact area will be, and therefore the stiffness, the effective stiffness of the cell will become larger, will be a function of loading force and tip geometry. In order to get the materials properties, we need to employ a model, and usually what is employed is a variant of the Hertz model, which has been invented or derived the first time here by Heinrich Hertz at the end of the 19th century. It has a lot of assumptions, and usually AFM people don't worry too much about these assumptions. If you think about cells, and if you are very strict, basically none of these assumptions is really fulfilled by cells. One of the assumptions, it's a homogeneous material. No, cells are not a homogeneous material. If cells would be homogeneous, so if the cell's mechanical properties would be the same at each position, there would be no use using the AFM for recording the mechanical properties, because you will get the same numbers all over the place. So it's very important for us that the material is not homogeneous. It's actually not isotropic because there's filaments in there which causes the stiffness. So this is also not taken care of. The most important point is actually the finite thickness of our samples. Cells tend to be very thin, like a micrometer, a few micrometers in the flat areas, the lamellipodia, even only a fraction of micrometers. And we apply forces and generate indentations which are roughly on the same order, like half a micrometer, micrometer. So the indentation is comparable to the thickness of our sample. This is difficult to tackle. There has been attempts in the literature to deal with that issue. What we usually do is we try to focus on the thicker parts of the cells to get not in trouble with this limitation. Again, this is a point where it shows that it's very important to be able to position your tip on a certain location in order to choose the thick or positions or locations on your tip. Just a technical remark. Since this is, people usually use an AFM either pyramidal tips or spherical tips. The models which are available in the literature is actually for a conical tip. Well, a cone is close to a pyramid, but it's not really identical to a pyramid. There is modifications of the pyramidal model, of the conical model, which takes care of the slightly different contact area of the pyramidal tip. For a reason I do not understand, this modification is not very well known in the community. It has been done by Felix Rico, Daniel Navaja's group, some time ago, and I really like to advertise it here because, well, the difference is not too large, but still, I would like that people are using actually the pyramidal model. I've added here the cylindrical model as well because it has some advantages. It has also been introduced and used by Felix Rico and Daniel Navaja's group maybe we can go later on into questions that cylindrical probes actually will have some advantages. Amazingly, despite all the criticisms I'm here saying that the assumption of the Hertz model are not really fulfilled or are not really given, amazingly, if you look at AFM data, and this is data on a living cell where we applied that Hertz fit, and this works actually very, very well. So despite all the critique you could use, the experimental data seem to follow nicely the experimental, the theoretical fit, except the point of viscosity, which I will come back to later. Okay, so, and since it's working, you can measure the, you can determine the mechanical properties of cells as a function of position, and you can create these colorful force maps or elasticity maps where you encode in color the stiffness, and you see the structure of the underlying cytoskeleton, basically the structure of the underlying cytoskeleton, which is true for animal cells, that the mechanical properties are mainly determined by the underlying cytoskeleton, the actin network. In other types of cells, plant cells, bacteria, this will be determined by the cell wall, but I will focus here only on animal cells, and therefore I can focus only on the importance of the actin cytoskeleton. Okay, so that was kind of the introduction. How can we measure cell mechanics? We measure it basically by recording force indentation curves and then analyzing it with an appropriate model. Why should we do that? Why is that important? The importance of cell mechanics as a tool to learn something about the state of the cell, or actually the state of the cytoskeleton of the cell, is an old idea. And it comes from Elliot Elson in a time, it comes, he did not use the AFM. He used the machine he called cell poker. But basically the experiment was similar than the AFM experiment. He had an indenter, was bringing that indenter in contact with the cell sample, indenting the cell, and then analyzing force versus indentation. His indenter was much larger than our AFM tips. Therefore, he could not investigate single cells, or he could not even go to certain areas of single cells. He had to investigate tissues or large clusters of cells. But from the science behind it, why do I want to measure cell mechanical properties? It was kind of the same story, and he, this paper here is a review he wrote in this paper here is a review he wrote in 88, so 1 year after the AFM has invented, 3 or 4 years before any person had ever looked at a living cell in AFM. He wrote already a review about the same topic. The topic of cell mechanics actually has picked up as AFM has become available, and as it has been shown by many, many people that you can investigate mechanical properties of cells, and it has been shown by many, many people that you can see differences in the mechanical properties of cells depending on cell type, or depending on state of the cell, depending on whether you're looking at a normal cell or a diseased cell. This is just 4 examples of a large group of papers which have been done basically on that topic. One of the earliest publications on this topic was actually already on cancer cells here by Malko Satalika in 1999. She already found then that cancer cells seems to be softer than normal cells. We will have to come back to that issue, why this is the case and what we can learn from that. There's other, and still there's an issue. It's not clear why the mechanical properties of cancer cells should be different than the mechanical properties of normal cells. If you think about other applications like here, healthy and infarcted heart cells, well, heart cells, they are generating forces, they are beating myocytes. You could imagine that there is a mechanical difference between healthy and infarcted heart cells. So it's obvious, or it makes very much sense to look at the mechanical properties of these heart cells. This is an example from our group where we're looking at the mechanical properties of the cell nucleus, which was also related to some disease which changes the lamin layer inside that nucleus. And there it was also hypothesized that the origin of that disease could be mechanical in some respect. Cancer cells. Many studies, as said, it started with kind of Malgorzata-Leka studies in '99. Many studies have shown that cancer cells are softer than normal cells. However, it's still unclear what is the physiological reason, what is the biological reason, what is the medical implication of that, despite that large amount of data which has been assembled. One point or one possible explanation could be cancer cells tend to be not as adherent as normal cells. So they tend to be adherent cells are very flat cells which are pulled down by actin stress fibers, and that generates force, and this makes cells always stiff. So one could argue that cancer cells are just softer because they have a different morphology. They are not as adherent, so they are more roundish. We were, some years ago, we were looking for cancer cells which are nicely adherent, cancer cells which do not show a different topography than normal cells, and we were happy to find them in a cell culture bank, actually at our campus here in Bremen. If you look at the force curves, again, the cancer cell here is much softer than the normal cell. So despite that the degree of adhesion or the morphology is pretty similar of the cancer and the normal cells in this case, still there is a difference in the mechanics. So it seems to be more than just the morphology. Oops, no, I wanted to show one more thing here. Okay, what you can see already from these force curves here, what we have plotted here is the curve recorded during approach and then the curve recorded during retract. Similar here for the cancer cell, the approach curve and the retract curve. And they are not identical. So the response of the sample is not purely elastic. We can analyze them with the Hertz model. We will get numbers. These numbers will be different for the approach and the retract curve. And strictly speaking, none of the two numbers will be right. The truth will be somewhere in the middle, because what we actually see is the elastic and the viscous response of the sample. Not only of the sample, because if you look at the part of the force curve here, before the tip actually touches the sample, there's a separation also between approach and retract. This is because of hydrodynamics. You pull, you drag your tip through the liquid, and there will be a hydrodynamic drag. And this is so in contact you will have a contribution of the hydrodynamic drag, but you may also have a contribution of viscosity of the cell. So you want to separate these two somehow, and you need to separate them. Before I come to the point how to measure the viscous properties, I just showed you before, you can see the viscosity in the data, so your sample is not reacting purely elastic. You need to develop a instrumentation or data acquisition system or analysis tools, which includes viscosity as well. But before making that technical parenthesis, I like to summarize kind of the current wisdom or current state or current state in the community on mechanics of cancer cells. It looks like that cancer cells are softer than normal cells when measured in the AFM. I have to add, when measured under standard conditions, which means on Petri dishes. This is kind of in contrast to what medical doctors say, because medical doctors say you can feel tumors in the skin or wherever, you can feel it by your fingers because they are stiffer. So a tumor seems to have different mechanical properties than the isolated cells, but this may be due to the extracellular matrix. You have seen that cancer cells and normal cells, they also have a viscous contribution. And it looked like in the curves I showed you before that the separation between approach and retract was larger for for the cancer cells. So it looks like that cancer cells are more viscous, but this is not really quantitative. We need to make that quantitative. This is just by looking at force curves, which is not really very scientific. What is still an open question is this of any cell biological relevance? Why are cancer cells softer than normal cells? Does that tell us something about cancer cells? There is one misconception in the community, one misconception which is kind of inspired of the behavior of red blood cells. Those who are familiar with red blood cells, they have to be very, very flexible because they have to squeeze through tiny capillaries, which are much thinner than actually the dimension of the cell itself. And it's essential that they can squeeze through that. And they are pushed by the bloodstream through these tiny vessels. So red blood cells, in order to move around, in order to find their way, they have to be flexible. The story, and therefore you could argue, well, cancer cells, if they want to form metastasis, they have to move around in the tissue, and therefore they have to be flexible. Well, the point is, there is nothing which pushes cancer cells. There is no bloodstream in the tissue. They have to find their way themselves. They have to squeeze themselves through tissue. So they actually have to generate forces. And in order to generate forces, they need actin, they need myosin, and this then will actually lead to stiffer cells. So since cancer cells are migratory, you could argue they have to be stiffer. Well, not all cancer cells are migratory. Some are living in a tumor and they are not making metastasis. So there may be a difference from the depending on the state of the cancer cells. What I just want to say is it's not clear why cancer cells are softer. This is still kind of a point which needs to be investigated. And there's no, up to now, there's no real theory which could describe that. It could just be a side effect because the metabolism of cancer cells generally is very, very different from normal cells. And just as a side effect, the cytoskeleton could also be affected. From a medical point of view, it doesn't make a difference. If cancer cells are softer always than normal cells, then you could use that as a diagnostic indicator, diagnostic tool, no matter whether you have a scientific explanation for that. If it is reproducible, and if it is kind of a general feature, then you could use that. So for biomedical applications, it may be interesting, even if we still don't understand what's going on. Okay. I was bringing you to the point that in mechanical measurements of cells, you should not only focus on the elastic properties, but also take into account the viscous properties of the sample. And therefore, I want to give you a brief introduction how to measure the viscosity of the sample. First thing, qualitatively. If you look at the force curve, this is now a force curve taken at two different rates. This is the rate we normally use in AFM. You see some hydrodynamic separation here between approach and retract, but not very large. This is a force curve on the same cell, same spot, where we were using a much higher rate, which we normally don't use. You see artifacts like ringing here of the cantilever because it's moved so fast. And you see that the hydrodynamic drag is larger, as expected. So from that separation, you could calculate the hydrodynamic drag, and you could subtract that from the force curve, get kind of a hydrodynamic drag corrected force curve. And this is now the blue line here, which nicely corrects here in the off-surface part, but leaves some fraction here in the contact part. And this separation here at the fast scan rate is actually larger than the separation here at the low scan rate, which would be acceptable, or which would be to be expected for viscosity of the cell. If you go even further down in rate, you would expect that the two curves sit on top of each other because the viscous response will become even smaller. However, cells have found ways around to escape the laws of physics. This is now the same cell recorded at very, very small scan rates, and you still see a separation between approach and retract even after hydrodynamic drag correction. Why? Well, cells are living, cells are moving around. So in principle, the experiment would work. Make the experiment very, very slow, and then you will get the elastic properties. Make the experiment faster, then you will in addition get the viscous properties. However, the cells are not as stable as we physicists would like to have them. So you have to find other ways to determine the viscosity. And there is two other ways. Andrea was actually introducing both already in her presentation, and we have used them both some time ago. One experiment is the modulation experiment, where you apply modulating force at different frequencies and measure the response of the cell to that modulating force. The other experiment would be a creep experiment, where you apply a jump-in force. What we have actually done here, this is kind of a force curve, but the deflection plotted as a function of time. So here we are off the surface, here we're ramping up the force as in the approach tract of a force curve. Here we stop set movement. There will be relaxation, creep relaxation of the cell. When we have achieved kind of an equilibrium situation, then you apply a jump and you see a well-defined creep during the loading and the unloading step here. Well, I will discuss that a little bit more in detail later on. So basically you have two options, a modulation experiment or a creep response experiment after applying a jump-in force. The modulation experiment we have done several years ago. As said before, it will be best to apply directly a force, which is not simple in the AFM because you don't have a direct force actuator. Well, in this experiment, we were applying directly a force because we were attaching a magnetic particle here at the very end of the cantilever. And with the help of a coil, we could really apply a force and look at the response. But basically, it turns out it works as good if you modulate the set height. And you basically get the stiffness of your sample as a function of frequency. In this particular sample, or in this particular cell, you basically get two different regimes. One, where the stiffness is more or less constant as a function of frequency. You are ramping the frequency from very low, 0.1 hertz, up to 1 kilohertz. And the other regime where the stiffness is increasing as a function of drive frequency. This is a typical behavior for like polymer samples, which shows something like a rubber plateau and something like that the viscosity, the elasticity changes with some power law depending on the frequency. And you can make theory. This is theory together with data on actin gels, so not on live cells, but on the polymers, which are the polymers you find in the actin cytoskeleton. And qualitatively, you see a similar behavior. At high frequency, you have that power law behavior that the elasticity scales with the frequency to the power of 3/4. You have that rubber plateau or that independent regime, and there is a crossover at something like 10 hertz or a little bit below 10 hertz, which is also the point where we see that change. We are not able to change up to now due to technical limitations in the AFM. We are not able to change the frequency over, like in this example, it's changed over 5 orders of magnitudes, or no, over 8 orders of magnitudes. We can't do that in the AFM. Therefore, we are still here in that transition regime where the power law we measure is not that 0.75, but rather 0.5. But you have that 0.5 also here in that frequency range. So it kind of makes sense. The important point is, the important piece of information is something's happening at that frequencies, like 3 hertz or 10 hertz, which corresponds to timescales of something like 100 milliseconds or whatever. So the alternative experiment, and we did that quite a lot, is that step experiment. I basically explained it already. This is now the graph where you also see the set signal, which is ramped during the approach. Then it's kept constant, except that tiny little step here, and then you retract your sample, and you see the creep after the approach, which is very, very large, and which basically is kind of a superposition of many, many tiny steps you did here during approach, We let that relax, and then after relaxation, or after it basically has relaxed, not completely, but to a large extent, then we apply that step and we analyze it. If we zoom in that step, you see that something like a exponential decay. You can make a model, you can simulate that. The most simple model, which will reproduce the observed data, is that standard linear solid model. So you model your sample as a spring in parallel to a combination of a spring and a viscous element, a dashpot. This spring here is the cantilever, which you also have to take into account. You can do the math, and from the data here, you can calculate then two spring constants of your sample and that viscous, that friction coefficient. What you can also, well, the spring constants you basically see also here in the graphs here, because at the very beginning you apply a jump in force and you see a large response. This is basically due to K1 plus K2, because if you apply a very fast step, that viscous damping, that friction coefficient, that viscous damping element will have an infinite stiffness. Then, after some time, you will have relaxed. There will be no stress in that part here anymore, and the elastic properties you see are due to that K1. So basically, from these two niveaus, the initial niveau and the final niveau, you can infer the two spring constants. The friction coefficient you don't see by looking at this graph by eye. What you see is that relaxation time, which actually tends to be friction coefficient by one of these spring constants. And relaxation time gives you also a feeling on whether you are if you compare the relaxation time of your system, which is somewhere in the order of 50 milliseconds, you can you can compare this with the time scale of your experiment. Either you make an experiment very, very slow, meaning the time scale of the experiment is larger than the relaxation time, or you make an experiment very, very fast, saying the time scale of the experiment is much, much smaller than the relaxation time. This distinguishes whether you see mainly viscous or mainly elastic properties. Okay, you can do the same thing with these little magnets attached to the cantilever. I actually would like to skip that one transparency to come right away to this one here. The model gives you spring constants. So this is kind of a way a jump back, because when talking about the force curves, I was advertising the benefits of using the Hertz model, because the Hertz model gives you materials parameters, elastic moduli, instead of force constants, which are a function of the sample, and the contact area, which is a function of the loading force. Now we are back to just measuring force constants, so we need to apply kind of the Hertz model as well, and you can do that from that force constants. If you assume that your contact behaves following the Hertz model, you can actually calculate the elastic modulus and the dynamic viscosity of your sample. You get two elastic moduli because you have two force constants there. So summary here is what do we get if we compare now modulation experiments, step response experiments, and other experiments which have been done with other techniques measuring the elastic and viscous properties of your cells. The modulation experiment basically gives you a frequency at which the behavior of the cells is changing. And that wasn't with the samples we were looking at, around 3 or 10 Hertz. The step response data gives you a relaxation time. This is the time at which the cell can change its shape. And these 50 to 100 milliseconds, if you translate that in frequencies, it's actually the same number, or the same order of number. So basically, the two experiments give you kind of the same information. There is an intrinsic time scale in the cell of 50 to 100 milliseconds, and you see that either as the relaxation time in a step response experiment, or you see it as a change in behavior of the rheological properties in a force modulation experiment. Using step response, this is the first time where you can really measure the elastic moduli of the cells, because you take into account the viscous properties, and you get numbers for the true elasticity, which are around a kilopascal, depending on the cell type. Could be a little bit less than kilopascal. We also have examples that cells have 2 or 3 or 4 or 5 kilopascal moduli. And you measure the dynamic viscosity, which the numbers are here on the order of something like 200 Pascal times seconds, which is actually a number people have measured for whole cells a long time ago using pipette aspiration experiments. So these numbers we get by AFM actually match very nicely what has been measured a long time ago with different techniques or setups. In discussing our step response data, in discussing why are we analyzing our data only in terms of the of a simple relaxation time, why don't we use more elaborated models like the power law rheology model I was advertising here for the modulation experiments? Well, what is the physics behind the power law behavior? The physics behind power law behavior is that you have several processes going on in the cell at different length scales and different time scales. Each of these processes has a associated relaxation time. And if you add together an infinite number of these different processes with different relaxation times, you will get a power law behavior. Actually, this theory has been developed in classes, and it has been applied to polymers. It is very important that you are in a regime where you have many different processes, many different relaxation times, and you look at the superposition of these relaxation times. In real life, there will always be kind of a fastest relaxation time, because the molecules can't move faster than a certain frequency, even at very small length scales. And there will be a slowest, a largest time scale. This is the time scale when the cell as a whole will change its shape, or its will change its shape. And we are actually in that relaxation experiments, we are at that end. We are measuring the slowest relaxation time, the slowest mode of the cell. So we're definitely not in a regime where we have a superposition of an infinite number. And that's basically the idea why we rather like to talk about relaxation times instead of power law rheology. Although we know that cells investigated at other regimes and frequency will show power law behavior. Okay, we can skip that. So that was the parenthesis on how to measure the cell's viscosity. I will come back now to where we started, like the cancer cells, to learn or to bring all together. So we started measuring the viscoelastic properties of normal and cancer cells. As usually cancer cells are plated on petri dishes, and we got numbers that the cancer cells, the normal cells, are much stiffer than the cancer cells. We also got numbers for the dynamic viscosity, which turned out to be larger for the normal cells also compared to the cancer cells. So it is not true what people have claimed, including us. We also have claimed that at some point. It's not true that cancer cells seems to be more viscous. That was inferred just by looking at the force curves. But if you do it quantitatively, you see the different story. So cancer cells are softer than normal cells on petri dishes, but they are not more viscous. They are actually less viscous. I'm pointing out that on petri dishes, because we were investigating the cancer cells also on softer supports, soft polymer gels, because a cancer cell in the tissue will be neighbored by other cells. So it will be in a surrounding which has a similar stiffness than the cell itself. In cell culture conditions, cells with stiffnesses around a kilopascal, they will be in contact with a Petri dish which has a stiffness of 100 megapascal, several orders of magnitude more. And as a matter of fact, the properties, the mechanical properties change if you change the stiffness or softness of the underlying substrate. So basically, we measured cancer cells and normal cells on a soft gel, which had a stiffness of only a few kilopascal, on an intermediate stiffness gel, which had a stiffness like 30 kilopascal, 10 to 20 times more than the cell, and then on the Petri dishes. It's easier to look at the averages we have gotten then. Let's look first on the left-hand side of that graph. This is the storage modulus for these three cases: soft gel, stiff gel, and Petri dish. So the stiffness or the elastic modulus of the cell is increasing with increasing stiffness of the support. This is actually something which is known for many, many cell types. This was no surprise. And we also see a change, if you look at the y-axis, this is the dynamic viscosity, we also see a change in the dynamic viscosity. Now let's look at the cancer cells. Same story, measured on a soft gel, stiff gel, and Petri dish. Here they cluster around pretty much the same value in terms of elastic modulus and in terms of dynamic viscosity. There is some scatter, but there is no real pattern. So the cancer cells, they seem to have kind of a stiffness or viscoelastic properties, more or less independent of the support, different like the normal cells. And if you put the two together, you will actually figure out on the Petri dish. So if you compare that blue one with the red one, on a Petri dish, the cancer cell is softer than the normal cell. On a soft gel, so if you compare this one, that's the normal cell, and this is the cancer cell, it actually reverses. The cancer cell is stiffer than the normal cell. So it is not true in general that cancer cells are softer than normal cells. Cancer cells are softer than normal cells on Petri dishes. On soft gels, the behavior could be different. More importantly, the viscoelastic properties of cells do not change when you, or do not change very much, when you change the stiffness of the underlying substrate. And this is different from normal cells. So in some sense, the mechanical phenotype, the way how cells sense and react the stiffness of the underlying substrate, this is very different between cancer cells and normal cells. So it's not the point that accidentally cancer cells are softer than normal cells makes them different from normal cells. It's more general. Cancer cells don't care, don't react on the mechanical properties of the underlying substrate. Normal cells do. And this is something which has also become, has attracted some attention in studies outside the AFM field. What you can see here is that if you compare normal cells, benign cells, with cancer cells of low metastatic potential or cancer cells of high metastatic potential, you can see that these benign cells, they are not interacting with the underlying substrate, but the other ones are. They try to invaginate the underlying substrate. They try to migrate through the underlying substrate. They try to make metastasis. In order to do that, they have to not adapt to the stiffness of the underlying substrate as normal cells do. They have to fight against it. So this may be actually the biological explanation. Why is the mechanical properties of cancer cells different than the mechanical properties of normal cells? It is the fact that cancer cells are cancer cells and they want to form metastasis. Okay, I come to the end of my presentation. The summary on the cancer cells is the real difference as it looks to us, the real difference between normal cells and cancer cells is normal cells adapt their stiffness to the stiffness of the underlying substrate to some extent. Cancer cells don't do that. The important question is that a general feature? Up to now we have, well, I've shown you data of one sample of a pair of normal and cancer cells. We have studied a few more up to now, and we have seen that this pattern actually seems to be repeating. If this pattern repeats, then there is kind of a crossover. If the support is very, very soft, then the cancer cells will be stiffer than normal ones. If the support is very, very stiff, then the cancer cells will be softer. So there will be kind of a crossover at some certain substrate stiffness. This probably, we don't know yet, this may change, may be different from different cell types or cancer types. Still, we don't know whether that's related to cancer cell function, but in some respect it makes sense because cancer cells do want to form metastasis, therefore they have to migrate through the tissue. General summary on the cell mechanics, because cancer cells was just one example for measuring cell mechanics. General summary is you can measure with the AFM the viscoelastic properties of the cell, not just only the elastic properties, and it's important to also record the viscosity, either by using step response or modulation experiment. The cell mechanical properties you are observing will be a function of the substrate, well, chemical composition, stiffness, possibly topography. It definitely, cell mechanics serves as a marker of the state of the cells, and this gives you, and this opens the possibility for applications, many applications, cell biology and possibly biomedical applications. And cancer cells was just one example for that idea. And in cancer cells, we see a difference between the mechanical behavior of the cancer cells and the normal cells. Okay, I was presenting you a lot of data. I want to thank all the people who were involved in recording that data. Just a few names, since these are current members of my group and still active on that topic. Camilla Riana, Holger Doschke, Akshu Yanku, and Prem Kuma. I want to thank you for your attention. I hope I could present you some new ideas, or could convince you that including measuring viscous properties in cell mechanics actually opens up a new field and is worth doing it. So thanks very much for your attention, and with this I want to conclude the presentation, and I will be happy to answer questions. So I think Bede will take over as a moderator now, right?
Yes, thank you, Manfred. That was a great talk. So we've got a bunch of questions here, actually enough that we will definitely not have time to answer them all, but we will get back to people through email on the ones that we do not have time to answer. But we will go ahead and answer a few here. The first one, Manfred, is for you. Basically, when you measure cell stiffness on different substrates, how do you take into account that some cells can be round and others are flat?
Well, this is an important point, that if you look at cells at softer substrates, they will not be able to be as flat as on stiff substrates, because in order to be flat, they have to be well adherent. And in order to adhere, they have to generate force and they have to pull them down. And this doesn't work on soft substrates. So generally, you will find cells on soft substrates to be less thick or less adherent than on normal cells. This is just the point that the cells are adapting, are changing, are interacting with the substrate. This is basically the reason why cells tend to be softer on softer substrates. So we can't really take that into account. Cells on different substrates can be very different, and therefore the mechanical properties will also be very different.
Okay, great. And another question for you. If the viscosity or viscous effect is dependent on frequency or rates, is it an inherent or intrinsic property of the cell, or is it a property of the measurement?
It is both. I didn't go into the the details actually, what you see here in that creep response experiment is kind of the apparent relaxation time, which is the relaxation time of the of this mechanical circuit. And it also depends on the cantilever spring constant. So the apparent relaxation time is a function of the cell and the experiment. But if you do the math, then you can actually calculate from the apparent relaxation time, you can actually calculate the intrinsic relaxation time, which turns out not to be too different from the apparent one. So you can analyze your experimental data such that you really get the intrinsic relaxation time as defined here, as defined friction coefficient divided by K2. And this tau, as calculated following that formula, will not be the same tau which shows up here in the fit. That's details. For those who are interested in the details, we are in the process of, or we actually have submitted a publication on that topic. There's an appendix. So if you're really interested in that, you can go through the equations in the appendix.
Thank you very much. Okay, so I have a question for Andrea here. What do sharp tips measure on the cells in contrast with the spherical tip?
Okay, so you heard Manfred refer to colloidal probes, and a lot of people use these types of probes. They're essentially an AFM cantilever with a glass sphere or polystyrene sphere on the end of it. It's typically used for a couple of reasons, in that it actually produces a really nice, well-defined contact area for when you're fitting your models for calculating modulus, as well as cells, when you're pushing on them with tips, they don't really like a lot of pressure. So sharper tips apply a lot of local pressure, and you can actually puncture through the cell membrane. So using spherical probes, you can actually reduce that pressure so you don't actually punch through the cell. Looking more at sharp probes, in terms of the images that we showed you of the microvilli and trying to get high resolution on the cell, you know, I guess sharp is relative. That was a 65 nanometer tip end radius, but, you know, sharper compared to a 5 micron bead on the end of a tip. So by using sharper probes, you can actually start to resolve some of the structural features on the surface of the cells.
Okay, and I'll take this one. The question is, have you tried to look at the elastic modulus dependence on probe vertical speed during force measurements? And the answer is that we've done this on gels a little bit, and we actually have a huge range of frequency that we can cover, because we have everything from force spectroscopy at less than a tenth of 1 hertz, all the way up to peak force tapping at around 1 kilohertz in liquids. And basically, so we have a lot of capability there, but there's a lack in methodology for quantitative treatment of that type of measurement. And we feel that for doing quantitative measurements, it's much better to use one of the 2 other methods that have been described during these talks, either the creep measurements, where you're holding the z position constant, or the force constant, or alternatively to do some kind of a measurement where you're modulating your tip or your sample in order to basically do a frequency-dependent measurement. Okay, and maybe one more here for Manfred. What would be an appropriate indentation speed to avoid the viscous effect when measuring elasticity?
To avoid the viscous effect of the cells? Well, yes. We have seen that typically we and other people ramp like the force curves at something like 1 force curve per second, which seems to be, you still see some viscous effect, but not very, very much. So you would like to go below that, maybe 0.5 hertz, but then actually what you figure out is that there is motion of the cell going on. The cells are moving around. So I can't really answer that question. There is no real good time scale for doing that. You have to be so slow that you pick up other processes of the cell, not just the passive response to the force you apply, but active processes going on within the cell. So I guess you have to live with kind of compromise, and that compromise often people use is like 1/4 curve per second, which gives you a little bit of viscosity, but not too much, and a reasonable degree of stability of your sample.
Okay, that's great. Okay, so I think with that, we'll go ahead and wrap up. We have quite a few more questions here, so we will be getting back to people with email. But thank you very much for your attention and for joining us. And thank you very much, Manfred and Andrea, for speaking.
Thank you.